All right then. Well, hello everyone, and welcome to our fourth Dean's Digital Cafe of Spring 2025. I'm glad you could join us today. I'm your host, William Letsu, Assistant Professor in the Biological Science Chemical Sciences Department at New York Tech. And on behalf of Dean Moore of the College of Arts and Sciences, I'd like to thank all New York Tech students, faculty, staff, alumni, and guests here today for tuning into our event. I'd like to remind everyone that today's talk is being recorded. We'll be opening the floor for questions and discussion at the end of the presentation, but you should feel free to enter your questions or comments into the chat at any time. And stay tuned for our new Dean's Digital Cafe webpage on the College of Arts and Sciences section of the New York Tech website, where you'll be able to watch recordings of today's and past events. So hopefully coming soon. Before introducing our speaker today, I'd like to acknowledge the land. The Long Island campus of New York Tech sits on a larger territory known as Pominoke, which was home to numerous indigenous communities, including the local tribes of the Matancox and the Merricks. I'd like to acknowledge the land, the stewardship and sovereignty of the native peoples, and their enduring importance, even in this virtual environment, to our work and community here today. And now it is my great pleasure to introduce today's speaker, Dr. Subhabrata, or Subha Chaudhury. Dr. Chaudhury joined the Department of Biological and Chemical Sciences at New York Tech in 2023, where he now teaches organic and pharmaceutical chemistry at the New York City campus. He received his PhD in organic chemistry from Marquette University and has held postdoctoral research positions at the University of Kansas, Medical College of Wisconsin, the University of Glasgow, and the University of Notre Dame. Prior to his joining New York Tech, Dr. Chaudhury was a visiting assistant professor at Marquette, and his research focuses on the development of small molecules as cancer chemotherapeutic agents. Today, he'll be telling us about his work on pyruvate carboxylase inhibitors. So welcome, Subha, and we are excited to learn more.
Good afternoon, and thank you very much, William, for your kind introduction. And and I really appreciate the faculty members, students, and the staff who has joined in here. So let me put that into okay, right. Okay, now can you all hear me without any problem?
You're coming in loud and clear.
Okay, thank you. So today I'm going to talk about a project which I have started a few years back. So as an synthetic organic chemist who has interest in medicinal chemistry, my research is very much focused on the target-based drug discovery. And in this case, our target is a metabolic enzyme called pyruvate carboxylase, which is not a very popular target for disease area because it's very much related to the cellular metabolism, and that's the problem. So you don't want to disturb the cellular metabolism as a whole, even though there are similarities between the cancer cell metabolism and the normal cell metabolism, and there are also differences. So you have to be very selective if you want to target pyruvate carboxylase, because the cellular metabolism is very much dependent on this particular enzyme, and I'll try to explain why and how it is related to cancer. Now, before I go into the pyruvate carboxylase, I want to talk a little bit about some of the facts. It's not really unknown to everyone that the cancer is a devastating disease. It accounts for the death amount of the US, only second to the heart disease. So just to give an approximation, why it's not really working? Okay, sorry, let's see, this pointer, laser pointer. Okay, so now cancer alone is reason for 600 deaths per day in the US, according to the American Cancer Society. So that's a lot of death. And cancer is not only causing the death, but it is devastating anyone and everyone associated with the cancer patient. So it imposes a big financial burden to the family of the person who got the cancer, and it got a very long and high impact on the society as a whole. So it's a menace to the society. Unfortunately, even though there are significant progress in last century on cancer research, but the reason is not fully understood. We know that genetic mutations are the reason, or if you have an oncogene present within the system. Other than that, the cancer causing environment or the habits or the lifestyle can also cause cancer. So, but actual reason is still not fully understood. So, urgent need for cancer education, early diagnosis, and continued research for cancer prevention is very important. So, in this regard, researchers have targeted a variety of cellular targets which are overexpressed during cancer, particularly the proteins, enzymes, chaperones, and signal molecules. So pyruvate carboxylase is, as I said, it's a metabolic enzyme. It's localized in mitochondria, and it got, in the active form, it got 4 identical subunits, meaning that it's a homotetramer when it reacts. And it catalyzes the transformation of pyruvate into oxaloacetate. So it takes up 1 ATP for the energy in this conversion. And I'll explain later how it actually works within the pyruvate carboxylase site. So before I do that, I just wanted to tell you that how important this pyruvate carboxylase enzyme is. So pyruvate carboxylase is not participating directly into the TCA cycle, but it supplies a very important precursor oxaloacetate. As I said, that pyruvate carboxylase catalyzes the transformation of pyruvate into oxaloacetate, and this oxaloacetate is replenishing the TCA cycle, and this TCA cycle intermediates are very important as they're supplying the precursor for the biomolecules. So the TCA cycle intermediates are actually the synthetic precursor for various amino acids, fatty acids, and glucose, even in case of normal cell. The other thing I would also like to mention that once the pyruvate carboxylase, so this pyruvate actually comes from the glycolysis process from the glucose, and then it's kind of fed in as an oxaloacetate by the pyruvate carboxylase. But if you have excess production of the oxaloacetate or even malate, then the reverse process, which is gluconeogenesis, can also be triggered. So you can also end up synthesizing more glucose if the pyruvate carboxylase is supplying excess oxaloacetate into the TCA cycle. So that's how it's also somehow related to the type 2 diabetes. Actually, this process in the normal cell happens once you are under the fasting condition. So pyruvate carboxylase in a very important enzymes, because it not only supplies the oxaloacetate, but it also sort of supplies the intermediates of the TCA cycle, which are important for synthesis of the important biomolecules, which might be needed in case of cancer once the cancerous growth is in progress. And I'll tell you the connection between these two. So this conversion of the pyruvate to the oxaloacetate is called anaplerosis. So this anaplerosis process is triggered in case of certain diseases, particularly in recent years. There are a number of different types of cancers which are linked to this pyruvate carboxylase, particularly the breast cancer, non-small cell lung cancer, as well as the thyroid cancer. And as I said, that the type 2 diabetes is also linked to pyruvate carboxylase. There is another disease which is a bacterial infection disease, where pyruvate carboxylase is also targeted, but that's not the human pyruvate carboxylase, that more of a bacterial pyruvate carboxylase is targeted. It's a bacteria called Listeria monocytogenes. So, but we are more interested about this area of research which is related to the cancer. So now, as I said, that pyruvate carboxylase um, uh, can really replenish the TCA cycle, which is also in turn responsible for supplying the precursor for the biogeneration of some of the biomolecules important for the cellular growth. And that's how it's related to the cancer, because in cancer you get uncontrolled cellular growth and you need those nutrients for the expansion of the cells. So cancer cells really rely on the ample pool of the TCA cycle intermediates for its rapid growth. The other indirect experiment which came in is this a knockdown experiment. So if you knock down the PC gene into some of the breast cancer cell lines, then it reduces the proliferation of some of the cancer cell lines. So this is kind of an indirect proof that PC is directly related to the cancer cell proliferation. Even though this experiments, this is not a real good way of studying the pyruvate carboxylase influence on the cellular metabolism, because if you silence the if you silence the corresponding gene, then you also deprive the other genetic products from that particular gene. So the best way to study the PC effect on the metabolism is making the inhibitors and then starting the pyruvate carboxylase influence on the metabolism, on the cancer cell metabolism. Okay. Okay. Now, okay. Now why we are developing this pyruvate carboxylase inhibitor. So first of all, as I think I already convinced you that pyruvate carboxylase inhibitors would be probably the best way to studying the influence of pyruvate carboxylase on the cancer cell proliferation. However, I mean, until very recently, we published our first, or actually the second paper. There was no potent or selective pyruvate carboxylase inhibitors in place. So what scientists have been trying are some of the non-selective pyruvate carboxylase inhibitors, and they only work in a very high concentration. Oh, why am I having? Oh, okay. They only work in a millimolar concentration, and they are not selective, meaning that if you use that kind of concentration, like sometimes like 5 millimole or higher, then that can also inhibit the other cellular process. The known non-selective pyruvate carboxylase inhibitors, which are currently being used by the researchers, are some of the ATP analogs as well as the pyruvate analogs, because these are very similar to the substrate pyruvate and the phenylacetic acid. So our project goal was to develop a potent and selective small molecules which will inhibit the pyruvate carboxylase, and for that matter, finally we will study the inhibitory effect of the pyruvate carboxylase on cancer cell metabolism. So what are the characteristics we are looking at? We are actually looking at three characteristics of these inhibitors. So first is the selectivity. As I said, that selectivity is very important, and we want to minimize the off-target effect. The potency, of course, is also very important, and the permeability. For the selectivity, probably for my students, the selectivity, I mentioned this is very important. So a selective inhibitor will only selectively target the protein. In this case, it will be pyruvate carboxylase. So there are a lot of other enzymes which also have homology with the pyruvate carboxylase, and I'll mention some of those, such as human carbonic anhydrase or matrix metalloprotease, or even like biotin dependent enzymes such as guanidine carboxylase. They're very, very similar in terms of their structure and functions, so they should not really get inhibited by that. So that would be number one. The number two would be the potency. So we don't want an inhibitor with having like a millimolar, working only at the millimolar concentration. So because that might also inhibit the other cellular process. That's a very, very, very high concentration, and the dosage will affect also, by that. So we would like to reduce the effective concentration, or more often the IC50 value, for this kind of inhibitors. So the potency would be one of the challenges. And the third criteria is the permeability. So the inhibitor should really go inside the cell by going through the cellular membrane, which is a bilayer. And not only that, after it gets into the cell, it has to go into the mitochondria, because I told you that pyruvate carboxylase is localized in mitochondria. So it has to really go through the cell membrane as well as the mitochondrial membrane. So these are the three challenges we had when we started this project. And obviously, our starting point was using some of the compounds which were similar in their functionality with the pyruvate. So pyruvate, if you just look at the structure, this is nothing but an alpha keto carboxylate, or if it's protein from the alpha keto carboxylic acid. So we found a bunch of cinnamic acid structure. I'm sure the chemists will know the cinnamic acid structure. So the cinnamic acid is nothing but the alpha hydroxy or alpha enol carboxylic acid. And the keto acids can really remain in an enolized form, and that would give you a kind of cinnamic acid structure. So we synthesized and we commercially we also purchased the commercially available cinnamic acid derivatives, and then we screened those compounds against a bacterial PC called Staphylococcus aureus PC, and we gathered those results, and it shows that one of the compounds, which is the alpha enol carboxylic acid, and you see the similarity in the functionality. So this keto in this case just got into the enol form, and this is the quinoline moiety. So this gave us a kind of a potency less than 10 micromolar. So it's a single digit micromolar concentration came in, which was a great, because it kind of gave us from a millimolar range to the micromolar range. So that's a good achievement. And when these enzymes were assayed against some of the very, very similar metalloenzymes, such as human carboxy anhydrase as well as matrix metalloprotease, they are not really inhibited up to a 200 micromolar concentration. So it got reasonable selectivity as well. However, when we tested the log P, that's the partition between the octanol water. So we had a range. So if it's -5 is a a very good permeable. -8 was the other end, so it's not permeable at all. So this kind of compounds were not really cellular permeable, so there was no cell-based assay was performed. And it's kind of understood that carboxylic acid, in particular the alpha hydroxy carboxylic acids, are not known to be cellular permeable. So that was the problem. So this was our first generation pyruvate carboxylase inhibitor, but at least we got a good potency and the reasonable selectivity. Only the permeability was the problem. So we needed a new class of pyruvate carboxylase with higher permeability. Now, at this point, we employed an in silico screening. So we had a collaborator from a company based in San Francisco called Atomwise, and they did a virtual screening using their AI-based AtomNet screening platform. And Enamine, as you probably know, that is a chemical company. They have a good database, so they screen just around 2.5 million compounds, and then after the first screening they got like around 30,000 compounds, and from there we filtered those 30,000 compounds based on the drug-like structure from Lipinski's rule of 5, as well as the pain's properties, and we identified 94 compounds which are reasonable, with a reasonable score for in vitro assay. So the in vitro assay was done in our collaborators lab at Wisconsin Marquette University, and after screening all these, we got a single lead compound, and we call it 2,4,5 imidazolidinone triones, where I will probably discuss a little bit more about the structure in the next slide. So here it is. So this is a 5-membered 2 nitrogen containing ring that's called imidazolidine, and then there are 3 carbonyl groups on alternative atoms on the 5-membered ring. So that's why it's trione, and both the nitrogens are substituted with 2 alkyl groups. So basically it's a disubstituted imidazolidine trione. We call it IZT, and probably in the future talks I'll just refer this compound as substituted IZTs or just the IZTs. Okay, now once we had our lead compound in hand, we quickly made almost like 200 compounds, and a very convenient 2-step synthetic route was just used for this synthesis. So we took isocyanates, various different isocyanates commercially available, and we reacted with various amines which are also available commercially to give us the corresponding disubstituted urea, and this was mostly the solid compound, and then the urea was reacted with excess oxalyl chloride to furnish this 5-membered ring. Okay, and this is our disubstituted triones. So the beauty of this reaction is that if you just use one particular isocyanate and you keep changing the amine, then you will get a series of IZTs or triones having different R2 groups, where R1 will be the same. On the opposite side, if you just keep the amine unchanged and change the isocyanate, then you will get an R2 unchanged and a different R1 groups. So that way you can make by permutation combination a lot of different compounds with different substitution on the 2 nitrogens. An alternative procedure was also I would say discovered in this case, because this reaction was not very well known. So we also found that there also monosubstrate urea, which are commercially available. And when we reacted with excess oxalyl chloride, that gave us the monosubstrated IZTs or the triones, and a Mitsunobu reaction could be used to furnish the R2 group here. For those chemists and know about this reaction, this is a very useful reaction, but generally the triones were never used as a substrate. So in that respect, this is a new reaction which are developed in terms of Mitsunobu reaction. So in Mitsunobu reaction, these alcohols are generally used as an electrophile, so the OH group became a good leaving group in presence of diisopropyl azo dicarboxylate and triphenyl phosphine, and then this nitrogen atom attacked as a nucleophile, and then you get this trione. So the different types of benzyl alcohols as well as the pyridyl alcohols were used to make these disubstituted triones or IZTs. So many derivatives, as I said, like almost like 200 compounds were prepared, and then we did the SAR, the structure activity relation, and what we have found that the compounds which are exhibiting a low micromolar value in terms of IC50 or highly potent compounds, like a single digit micromolar, are the compounds having R or one of the substituent as alkoxycarbonyl methyl, or you can say that ester methylene group on one side and an aryl or benzyl type or aromatic group containing a substituent on the other nitrogen. So it could be phenyl or benzyl or like thiophenyl or homobenzyl. So aromatic group is necessary for showing good activity, and that should be on one of the substituents on one nitrogen, and the other is the ester methylene group. So it doesn't matter even though it's ethyl or methyl. So our best compound, by the way, and this potency came around 3 micromolar. So even if we change the alkyl part of the ester from ethyl to methyl, it's still giving us a good potency. So the alkyl groups which are requirement was alkoxycarbonyl methylene, and on the other side an aryl moiety was necessary. So when these compounds were identified as good compounds, then we also tested them against other similar enzymes just to check whether they're selective enough, and it gave us a really good selectivity in this regard, and the permeability was also good. So the log p value was around -6 to -7, so which was reasonable. This is kind of summary what you got from our second generation compound. So the selectivity was good. So the additional metalloenzymes were also tried, and then biotin dependent enzymes such as guanidine carboxylase was also assayed against these compounds, and these compounds do not really inhibit those enzymes up to 200 micromolar range, so they're selective for the pyruvate carboxylase. And as I said, the permeability were tested against an experiment. It's called PAMPA, so parallel artificial membrane permeability. So that was evaluated, and it's reasonable. So altogether, substrate IZTs demonstrated high selectivity for the Staphylococcus aureus. So I probably already mentioned that these were all done against not the human pyruvate carboxylase, but a bacterial pyruvate carboxylase. So the bacteria is called the Staphylococcus aureus, and the reason being that they are highly homologous, particularly at the catalytic domain where the carboxylation happens. And I'll probably explain more on the domain in the later part. Okay, so yeah, so the compared to the first generation, the potency kind of remained the same. It's a single micromolar range. The selectivity has improved, and the permeability is also great. So now, yeah, so now I'm coming into the more structural detail. Now the question remains that where do these IZTs bind, because that can help us to identify the actual interaction between the drug molecule and the pyruvate carboxylase, and in turn that will also help us to generate the next generation inhibitors with enhanced properties, and also it will help us to hypothesize the inhibition mechanism, hypothesize the inhibition mechanism for that matter. So, as I said earlier, the pyruvate carboxylase in its active form stays at a homotetramer. So you can see, that's the active form of the pyruvate carboxylase. But if you just look at the monomer, so there are three major domains. This blue part, that is called BC domain or biotin carboxylase domain, where actually the carboxylase happens in presence of bicarbonate. So it just gets carboxylated by the bicarbonate at this domain in presence of ATP, and the ATP hydrolysis gives the required energy for it. Then there is a carrier domain where the cofactor biotin is attached. So this carboxyl group gets transferred into the biotin through this carrier domain. Okay. And then finally, this carboxyl group gets into the CT domain, which is the carboxytransferase domain, and that's where actually the substrate pyruvate binds and the carboxylation happens to give you the biotin. Sorry, to give you the oxaloacetate. So this is basically the mechanism. Now, when we did the kinetic experiment, since we didn't really have a good extracrystal structure, co-crystal structure to see or to find out where our inhibitors are binding, so we did some kinetic experiments, and it shows that it's a mixed type of inhibitor. So this is the Lineweaver-Burke plot, shows that it's a mixed type with respect to pyruvate, and it's non-competitive with respect to ATP. So ATP is binding to the BC domain. So the conclusion is that IZTs don't actually bind either at the ATP site or the pyruvate binding site. So it's probably binding somewhere else. Probably it's binding at the allosteric site. So so this is our hypothesis, and and I'll tell you why we are making this hypothesis, even though we do not really have a proof at this point. But our collaborators at Wisconsin, they got some low resolution crystal structures, and which shows that these kind of molecules, our molecules, the IZTs, bind at this binding site, which is in between the CT domain and allosteric site. Let me go back and then show you that there's a fourth domain which I was talking about. So yes, so this green domain. So apparently this doesn't really take part in the catalytic process. Whereas, I mentioned about this big domain, BC domain, this is BCCP domain, and this is the CT domain. But there is a green domain, this is the allosteric domain, and probably that's where it's binding, or it's probably binding at the in between the CT domain and allosteric domain. So no way it's kind of binding or disturbing the ATP binding site or the substrate binding site. So So it's probably binding at the exobinding site, and then it's preventing the carrier domain, which is the red domain, to access and lock the PC in its active conformation. So that's how it's kind of doing its inhibition job. Okay, so binding was predicted based on the low resolution crystal structure. So substrate ITs are potent, selective, and permeable, but that was based on the Staphylococcus aureus PC. And we already know that knocking down of the pyruvate carboxylase in breast cancer cells can reduce the cancer cell viability. So this inhibitor should, in principle, work in the human cells as well, because earlier we couldn't really do it in the first generation case because they are not permeable. However, once this enzyme, this inhibitors, the IGT inhibitors, were tested against the same cell line, the same breast cancer cell line, they did not inhibit those cells or the cell viability. So the reason could be of twofold. So first reason could be that substrate IGTs have a very low mitochondrial permeability. So because we only tested for the cellular permeability. So even though it's getting through the cell membrane, but probably it's not permeable to the mitochondria. So that could be the reason. The other reason, that since we are using the human cells when we are doing the cell-based assay, so maybe the IGTs do not really inhibit the human pyruvate carboxylase. So that is another reason for it. So we tested both the reasons. So in case of mitochondrial permeability, so we bypass the mitochondrial permeability. So we used brewer's yeast. So brewer's yeast, the pyruvate carboxylase is not really localized into the mitochondria. It's localized on the cytosol. So once we use this IZTs against this Breuer's cell, then in principle it should reduce the growth of the Breuer's yeast. And there is another known but non-selective pyruvate carboxylase was used, that is called L-aspartate. It's an amino acid. So it should be or both of them should be reducing the growth of this Breuer's cell. So this is the positive control, positive control and negative control. So L-aspartate does really reduce the growth of the Breuer's yeast, which is a known non-selective pyruvate carboxylase inhibitors. So what we got is in here. So unfortunately, our molecules did not really inhibit the growth of the Breuer's yeast cell lines. So the mitochondrial permeability was probably not the issue in this case. How about in terms of the human PC? So is that the reason that because it's, even though it's inhibiting the bacterial pyruvate carboxylase, but human pyruvate carboxylase is a different kind, so maybe it's not really inhibiting. And why don't we use the human pyruvate carboxylase? Here is the problem. So human pyruvate carboxylase unfortunately is insoluble using the bacterial expression system. So all efforts to prepare this human pyruvate carboxylase or synthesize this human pyruvate carboxylase have failed. So we devised something new. So when we chopped off or removed the BC domain of the human pyruvate carboxylase, then it was soluble. So meaning that the truncated human pyruvate carboxylase without BC domain was soluble, and then that was taken, and then we prepared a chimeric one where the BC domain was replaced by the human BC domain was replaced by the bacterial or Staphylococcus aureus pyruvate carboxylase BC domain. So we call this thing chimeric ones because you have all the features of the human pyruvate carboxylase except the BC domain, where the BC domain came from the bacterial pyruvate carboxylase. And this is the hypothetical chimeric human PC tetramer. And fortunately, these were all soluble and these were isolated and then tested. Now, if they do inhibit our compounds, then and our hypothesis said that BC domain probably doesn't have much role in binding the IgGs. So it should really give us more understanding of where our compounds are binding to these protein domains. Okay. So now this is the initial picture when the IZTs, so these are actually the IZTs number. IZTs, when IZTs were inhibiting the Staphylococcus aureus pyruvate carboxylase, and this is the results when the same IZTs were used against the chimeric human pyruvate carboxylase. So as you can see, the substrate IZTs are significantly less potent against the chimeric human pyruvate carboxylase. So then we went on to find another pyruvate carboxylase which got more homology to the human pyruvate carboxylase, and that is basically the pyruvate carboxylase extracted out of the pig liver. So the reason for having the pig pyruvate carboxylase is the following. So pig pyruvate carboxylase are physiologically, genetically, and metabolically very similar to the human pyruvate carboxylase. It got over 97% homology to the human pyruvate carboxylase. And the third reason is that pig livers are widely available. So our collaborators at Wisconsin, and they have butcher shops available. So this is the graduate student from our collaborators lab, Nick Snyder. So he got a bunch of pig livers from the local butcher shop and then extract the pyruvate carboxylase and then run those reactions in presence of pig liver PC. So again, this is the original or earlier results with the Staphylococcus aureus PC, and this is with the pig liver PC. Interestingly, they look very similar to what we got when we attempted the same kind of experiments against the chimeric human PC. So both the pig PC as well as the chimeric human PCs are not really getting inhibited or significantly not getting inhibited by our IZTs. So that's not a very good news, but we just needed some explanation for that. So yeah, so they're significantly less potent against this last two pyruvate carboxylase. And as a whole, we have also checked some other non-selective inhibitors, and it's found we find out that as a whole, the small molecules inhibitors are not very potent against either the pyruvate PC pyruvate carboxylase, sorry, pig pyruvate carboxylase or chimeric human pyruvate carboxylase. So what could be the reason? So there was one difference in the experiments. So the difference is that when you use the pyruvate carboxylase extracted from the Staphylococcus aureus, so that does not require any acetyl CoA during the experiment. However, once you use the pyruvate carboxylates extracted from the pig liver or the chimeric human PC, they both require the acetyl-CoA for their activity. So without acetyl-CoA, they don't really work. And acetyl-CoA are allosteric activators of the pyruvate carboxylase. So that could be the reason that we did not really use acetyl-CoA when we experimented with our bacterial PC. So what if we first activate our pyruvate bacterial pyruvate carboxylase with acetyl-CoA and then run the experiments? Would it make some difference? Actually, it did. So what we have seen that in presence of acetyl-CoA, even though it's the bacterial or Staphylococcus aureus PC, it does not really give you the same kind of potency or the inhibitory activities as without the acetyl-CoA. So small molecules inhibitors are less potent in general, not just the IGTs. In general, they are less potent against the acetyl-CoA activated pyruvate carboxylase. So therefore, small molecules inhibitors are universally less potent when they are activated. It may not be acetyl-CoA, but acetyl-CoA is needed in case of human pyruvate carboxylase or humanized pyruvate carboxylase. So the alternative approach should be that we use all the time that acetyl-CoA once we'll be testing or once we'll be assaying this kind of inhibitors. Without that, whatever IC50s we are getting, those are erroneous. So that was a good learning. And we also know that our pyruvate carboxylase inhibitors, the IGTs, are not really binding either at the BC or CT domain, but it's probably binding at the allosteric domain, most probably at the juncture of the two domains. Despite these shortcomings, these substrate IGTs represent the best class of pyruvate carboxylase inhibitors due to their potency and permeability. And this is one of our best compounds. I probably showed you earlier. So this got an IC50 of 3 micromolar without the acetyl-CoA, of course. Okay. So yeah, these are the conclusions. So substrate IGTs are the best class so far. And the paper we have published, last year in 2024. Yeah, our hypothesis is that the substrate IGT is binding at the exo binding site. So it's not really directly binding at the CT binding site or the BC binding site. And probably it's preventing the carrier domain access to get the carboxylic group into the CT domain, and that's how it gets locked into an inactive conformation. And small molecules are less active once it's activated by acetyl-CoA. So currently we are working on developing the inhibitors for allosteric domain, and we'll of course disclose our results in the due course. So we are not really targeting anymore either the BC or CT domain. So our target is more on the allosteric domain. Okay. So yeah, before I end this talk and open the floor for questioning, I'd like to thank immensely to my collaborators, particularly Martin St. Marius at the Department of Biological Science at the Marquette University in Wisconsin, and Nick actually did all the enzyme assays I showed you, and now he's Dr. Nick Snyder. So he moved out of the lab. So he was his graduate student by then. And the initial chemistry was developed in Dr. Donaldson lab when I was a postdoc. But currently, I'm just working more on a new scaffolds to target the allosteric domain. and these are at the very preliminary stage. And but we are getting some interesting results, and probably I'll be able to share some good results in coming days. And the Neil Hendrickson was our collaborators who did all the in silico docking and molecular modeling. And we are very thankful to for the NIH support. Gives us the R15 grant. And I thank you all for your kind attention.
All right, thank you very much, Subha. That was a great talk. Do we have any
Thank
questions
you.
from the audience?
Right. Hi, Subha.
Hi.
Very, very nice talk and very clear, very elegant work too. So thank you for sharing that. I just have a conceptual question. So pyruvate carboxylase, right, plays, as you indicated, plays a key role in gluconeogenesis, and other processes such as anaplerosis. But and cancer cells are not the only cells that are doing this, right? And actually, those two processes are very important, say for liver cells, for pancreatic cells, etc. How do you, so let's say you come up with a great inhibitor that now is able to inhibit PC. How do you plan to eventually target that specifically to target cells so that you will spare all the other metabolic cells that require these processes for normal cell functioning?
That's a very, very important question, and I really appreciate. Yeah, that's actually a concern, and that's why probably people did not really target pyruvate carboxylase, because it will not only affect the cancer cell, it can also arrest the TCA cycle of the normal cells, right? What we believe is that there is a significant difference in terms of structure or activity of the cancer cell pyruvate carboxylase from the normal cell pyruvate carboxylase. That's our initial hypothesis. So if we have some inhibitors that will probably selectively bind to the cancer cell pyruvate carboxylase or the activated complex of the pyruvate carboxylase, because in some cases what we have seen that the pyruvate carboxylase is either overexpressed or it's hyperactive. So meaning that it's not necessarily the pyruvate carboxylase concentration is higher in some cancer cells, but it's super active. So maybe our inhibitors will go and selectively bind to those more activated or hyperactive pyruvate carboxylase which are localized in cancer cells. That's our initial hypothesis.
Thank you, Subha.
And yeah, I mean, your question is very, very legit. And at this point, maybe you have seen that in my title, I tried not to say that we are making a chemotherapeutic agents for cancer. We are saying that we are actually developing the molecules for studying cancer cell metabolism. So at this point, at least we can do some metabolic studies that how pyruvate carboxylase is affecting the cancer cell metabolism. So once we have more information, whether the pyruvate carboxylase from the normal cells are different or more activated, then we'll probably be able to make more selective inhibitors which will target only the cancer cell pyruvate carboxylase but not the normal cell pyruvate carboxylase. Does
Perfect.
it
Thank
answer
you,
your
Subha.
question? I don't know.
100%, yes. Thank you.
Yeah. Hi, Subha.
Hi.
Thank you very much. This is an excellent talk, and it's complex work. So
Thank you.
Thank you very much.
Thank you.
I mean, I'm following up on the question that Henry was just asking is how easy it is to chemically or attach these inhibitors to some antibodies, because the reason why I'm saying is that I believe that some of the cancer cells, they have their specific markers. So if there is an antibody for that, you could attach your molecule to these antibodies so you can make them selective or specific for the cancer cells. So my question again, how easy it is chemically to attach these small inhibitors to some antibody, so you can get an antibody that's a target for the cancer cells and combine them together, and that way you make them more specific for the cancer cells.
That's a very, very good question. You are talking about the bioconjugates, right?
Yes.
Biologics, bioconjugates.
Yeah.
Yeah, I mean, in this case, as I said, that this is not a very popular target, and we are at the very initial stage. But yes, that would be a very well therapeutic, and that might answer, what Henry asked. So if
Yeah.
it could specifically target the cancer cells, because you are using the antibody based drug target, so you can just use your small molecule as a weapon, and then the antibody will be your warhead, right?
Yeah. Yes.
Your missile, and the warhead would be your IZTs or any other. Yeah, so that is something we'll probably explore. And it's all about the chemical synthesis or the kind of functional groups which will be pendant at the molecule, and we have to really find out some chemical protocol to attach that to the antibody. So yeah, I don't really know any work that has been done. As I said, that there are not too many known potent and selective pyruvate carboxylase at this point. But definitely that would be a very good option. And I think you very nicely answered some of the questions we are wondering about, that how can this be making more selective towards cancer cells. Yeah.
Yeah, I mean, I think that should be your future strategy. But again, the only thing you would still need to redo your testing again, because I understand that attaching it somehow you may lose some of the activity or some of the properties. So you are going to have to, once you are moving forward with that strategy, you will have to do a lot of testing again, making sure that your molecule is still as active as it was before conjugation.
That's right. Yes,
Yeah.
exactly. So those will be probably the future work. And to be honest, you see that we are even at this point digging around the micromolar range. We didn't even think of the nanomolar range, and the reason being that we don't really have much high standard at this point, and not we don't really know how there are groups which are who are working in this area. So there are a lot of biologists who are working, but not too many medicinal chemistry groups who are working in developing this kind of inhibitors. So but at some point, I think we'll be able to bring it down to, say, nanomolar range. So once you have more potent and selective and probably permeable inhibitors, then we'll definitely think in terms of delivering those more selectively to the cancer cells.
Yeah.
So,
yeah. Thank you.
Yeah, thank you.
So I know we have some students in the audience. Do we have any questions from students? We don't always want to hear what you have to think.
Yeah, please, students, go ahead.
No. Well, I have one final, maybe I have one final question, Zuba.
Sure, go ahead.
Yeah, so the acetyl-CoA activation, have you looked at why that, like in terms of the shape of the molecule of the enzyme, why that affects the potency of your drugs? Does the binding of acetyl-CoA like change the conformation of the protein in any way that prevents it from binding and inhibiting?
That's a very good question. So all we know that acetyl-CoA is an allosteric inhibitor, and as far as I know, that there is no crystal structure available, or there is known crystal structure of the pyruvate carboxylase with acetyl-CoA, but it's not really competing with the substrate or ATP. So that's why it is hypothesized that it's not really binding at in those domains, but probably binding at the allosteric site. So what you were asking, that whether it's kind of changing the conformation so that the IZTs or any other small molecular inhibitors are not really binding to that, and that's what I think, that's what we think, or that's our hypothesis, that once the acetyl-CoA is binding to the allosteric site, it's bringing in some conformational change to the pyruvate carboxylase. So now it's probably not strongly binding to the IZTs, or maybe it's somehow blocking the IZT binding site at the allosteric domain. There could be two reasons, right? So either it's kind of changing the conformation, or it could be just blocking somehow, probably not completely, but partially blocking our inhibitors
Great.
to the binding site, because both of them, both the acetyl-CoA and our IZTs are probably binding at the allosteric site, or that's the hypothesis at this point, going for the same kind of site.
Right, makes sense.
Yeah.
Okay. All right. Well, if there are no other questions, well, thank you, Subha, and thank you all for once more for joining us today. Please join us again next week when Professor Michael Izadi from the Department of Humanities will be giving a very topical lecture on U.S. citizenship. So take care, everyone, and have a great rest of your day.
Thank you, everybody.
Thank you, William, and thank you all. Have a great day. Bye.